Thank you, Shahar, and good morning, everybody. It's a great pleasure to be in this forum. This is something that I have started when I was a postdoc at the Weizmann Institute and continues till today. And I truly appreciate the opportunity to discuss with you wide variety of sciences and technologies. Today, my talk will involve several terminologies, including chemistry, nanotechnology, advanced materials, medicine, artificial intelligence. So it will be really multidisciplinary in this endeavor. And therefore, I would like to speak a little bit more in lay term. So in many instances, and where is needed and where we can deepen inside the advanced materials of those fields that relates to the IVS, I will deep more inside. So practically, everybody has heard about a disease, and the term of disease means that we have a disorder that deviates us from healthy status. This could be discovered at early stages of the disease when the disease starts to emerge really in very short terms. If we catch up the disease at those stages, at the early stages where we still feel healthy, everything is fine and the treatment is quite easy and the survival rate is quite high as well. The other issue is that if we wait more time and let the disease progress to advanced stages, then we will have many problems and challenges. One of these challenges is the misdiagnosis, and the other one is the fact that the treatment will start to be much harder in the sense that can be done, the treatment can be done, but the survival rate will be low and the efficacy of the treatment will be quite low. And therefore, our wish in this field and our vision in this field is really to find a way to diagnose the disease quite early when the person still at the very early stages of the disease. Could you please mute yourself? In order to do so, we have to develop a device and a technology that can fulfill simultaneously several parameters. And the first one is the easiness of use. The other one, that the device has to be small and portable so everybody can use it and can be accessible to every community and to every home and to every clinic. And of course, the device has to be also a non-invasive. It's better to do it non-invasive so we can eliminate inflammations and to eliminate complexities in operating the device. And of course, the most important part of that technology or that wished device is to be highly accurate. Highly accurate means that if you are healthy, it will predict that you are healthy. If you have a given disease, it will say that you have a disease with really high accuracies. High accuracies in our terminology above 90%. There are many approaches worldwide how to fulfill this vision. And our approach relies on a hypothesis that was used 2,400 years ago by the ancient Greek. And the hypothesis says that when a disease starts to emerge inside the human body, such as the red spot, which you see here, this disease starts to emit volatile organic compounds, namely compounds with high vapor pressure into the blood. And from the blood, those molecules has to emit to the environment. And one way to do it is throughout the exchange with the alveoli in the lungs. And once they do so, they appear in the exhaled breath at very low concentrations that range between few ppbs up to tens of ppbs part per billions. Just to note that these are very low concentrations for wide variety of sensors to detect them. With this in mind, of course, this was a hypothesis and many people throughout the history have thought about it, but never was a real fundamental science behind it or a proof about this hypothesis. And this is one of the major challenges that we have taken for more than 10 years in my research group to see or to validate this hypothesis. And in order to do so, without going into all the details, we have went into 19 clinical centers worldwide with more than 15,000 patients and healthy controls with more than 17 types of different disease, whether cancer, kidney, whatever you find here also in the different organs of the human body. And we have collected their breath and we have analyzed their breath in a technique that is called mass spectrometry, and more specifically in gas chromatography mass spectrometry. And what we have found there, we have found that a set of molecules that we call it volatile organic compound with a number. Of course, this has a chemistry and it has a compound. And we have found a set or a pattern of molecules that appear in the exhaled breath of a person with a statistically significant ratios that could distinguish the healthy person from a person that has a disease. Also, what we have found that for every disease, the combination of these molecules that are, again, statistically significant, without showing the results right now, could be differ from one type of disease to the other type of the disease. For example, you can see that if we use one molecule, such as what we see here in VOC number 1, it cannot distinguish alone between different types of disease. Also, if we use VOC number 13, it cannot diagnose or differentiate between different types of disease. But nevertheless, if I take the whole combination of the 13 compounds, the whole combination will differentiate between different types of disease. And therefore, what we can claim, or what we have claimed and what we have shown in a field that is called today volatilomics, is the fact that for exactly as if every person has a unique fingerprint that could distinguish it from the other person in the room or in the world, this is exactly what we have in breath. There is a unique breath print that could distinguish one person from another person and one disease from other disease as well. Of course, more about it you can read in our published articles. But nevertheless, this device has proof was done with mass spectrometer, which is a great lab tool. But nevertheless, it has many challenges to be used as a point of care device. And one of these challenges is that the device is quite big, expensive, and slow in terms of operation. And also there is a complexity with the analysis of the results. And therefore, our challenge in the next step was how to engineer or to engineer our fundamental science into a device that could be a small, portable, simple, easy to use, cheap, and fast device to detect and differentiate between the different breath prints of the various disease which I have told you about from breath without any invasive issue. Therefore, we have developed and imitated an approach that imitates the olfaction system of the canine, in the sense that this device can collect the biomarkers of the disease from the exhaled breath, and then we use artificial intelligence or machine learning in order to get the results into specific patterns that can be used as our big data, with which we can compare new data and determine the disease. More specifically, what we have developed is, or the idea was to capture those molecules which are emitted from the disease into the exhaled breath with a set of array of sensors that are based on nanomaterials. And once they are on the device, the chemical interaction or reaction will be transferred as electrical signal to a machine learning operation system. And with that, and after a training process, we can get the status of the health, and we can get also the stage and more specific details about it. This is the whole idea. But in order to bring it into practicality, we have to do 4 steps. One of these steps, of course, as I have told you earlier, is to determine what are those molecules that appear in the exhaled breath of a given disease. And this is something that we do with mass spectrometry. And based on that, we go to the lab and we design nanostructures, and we modify the surface of these nanostructures by self-assembled monomers or by wide variety of receptors in order to increase the affinity of the biomarker, which we have found here with the nanostructures, which after then, these nanostructures we integrate into electronic device or with the electrodes that either as a transistor or as a chemoresistor. And what we do further, because we don't work with the selective approach, we produce many of these devices where every device differs from the other device in the array by it having different surface chemistry. Sometimes we modify the surface with given receptor. The other device might have the same core of the nanostructure, but the self-assembled monomer on the surface will have different functional groups in order to detect and increase the affinity to other types of molecules. So once we build an array of sensors, we connect it with a software which relies on machine learning. This is the first step. And in this case, we get a hardware that can do nothing actually. The next step in order to enable this device to serve as a diagnostic tool is to train the device. One of the ways to train this device is to take these simulations to produce these volatile biomarkers in the lab and their mixtures, and to expose it to the sensors array. Once we do so, we start to accumulate patterns of electrical signals. We store it in the memory of the computation system. And then when we have blind sample, we just make the comparison statistically. And based on this comparison, we can detect whether the person has a given disease, cancer, healthy, what kind of disease, etc., etc. And we can distinguish different types of cancer, stages of cancer, and more and more. As an engineer, back in 2009, we have finished the first generation of this system, and it was successfully implemented in the lab with a 93% accuracy. But nevertheless, when we have decided to take the same device with the same technology into the hospital in the same city again, the accuracy, which was 93%, dropped down to 55% accuracy when we have measured real human people. And then I have understood that the reason for this difference, that in real clinics and real world, we have much more heterogeneity between people, between samples, and we have many confounding factors in the environment and in the clinical states. And therefore, in order to validate devices that has to go to the industry or to the public or to the commercialization, we have really to validate it in clinical settings. And this is what we have done since then. And this is a new field that I have integrated with my engineering skills as well. Of course, today we do all of these 4 stages. And today what I will show you are the final results of clinical studies in order to be sure that whether the device works or not. The good news that after we have learned where we have failed, in the clinical studies, we have done many more research and we have succeeded finally. The most important part of this device is actually which structures we use in order to detect and capture the molecules in the breath and to transfer the chemical interaction to or interaction to electrical signals. One approach is to use chemoresistors that are based on gold nanoparticles, which are coated with organic ligands, which you see here, where we put a thin layer, this is TAM image, between the electrodes. And based on that, we measure the change in the electrical signal as a function of the interaction with the breath. Of course, as you can see here, we have different chemistries, and therefore the approach which we use, we interact the same sample in the gas with different sensors, and we measure the differences of the signals that are received between the various sensors. So this is a little bit different approach than the selective sensors that we are used to. The other approach, which has also complementary strength, is to use random network of carbon nanotubes that are linked between electrodes and are coated with organic ligands, and the same approach is being done here. Of course, as you can see in the example of the nanoparticles, we have an array of sensors you might see here on the screen. And in this case, each sensor, which you see by the different colors, react differently, as you can see by the thickness, to the same breath. And therefore, the whole signal and combination of signals between before exposure and after exposure will be different. And based on this difference, we do our analysis and we train our system as well. Of course, all of that today we do it on a piece of chip, which do the work quite nicely. But not less important, we have done a lot of work with many companies, with many institutes and many SMEs throughout European consortiums. And the result was to take a device, a lab device, which has a size of 1 meter by 1 meter and really heavy device, even though we have used nanotechnology, and to reduce it to a device that you see here in the screen, which you can use it in your or handle it. And this device allow the breath from a distance of 1 to 2 centimeters without intervention of any contamination from the environment. The device will pump throughout a micro pump the breath into a microfluidics, which host the sensors where the reaction will be done. And from there, this device is connected to throughout ICT technologies, and we get the results as I have explained earlier. And the most important part of this device actually is the artificial intelligence or the machine learning, because again, this device has to be trained. And therefore, we train it on different populations. We store the data, and the blind analysis will be, of course, compared with the memory that we have. This is something that we know with mass spectrometry, actually, but today we do it also with this very miniaturized device. It's very important to note that one of the most important parts of this device is when you capture the breath, because you have the so-called dead space and alveolar breath, and we need to take the breath from the lungs in a non-invasive way. And therefore, the timing when to detect the device is really very important. And therefore, there is a very small piece of sensor here that measures humidity and CO2 from exhaled breath with very high accuracy. And this is actually a development of one of my spin-off companies that I have co-founded, Nanovation, which really do this job quite nicely. With this device, we have went into clinical studies because this is our real validation. And we have found that it can distinguish between people, between diseased people and healthy people in quite high accuracies. And also it can distinguish between different types of disease that are within a category of 17 types of disorders. For example, you can see the red colors indicate that we have an accuracy of discrimination of more than 90%. And therefore, you can see that in most cases, we do get good separation between the types of disease based on exhaled breath only. In a few instances, such as in the green color here, we fail to make a discrimination. And this is a challenge that we will work on in order to compensate in the next stages. Because we have taken many patients from many countries, in this case, there could be a claim that the differences that we see here that indicate that we can discriminate from breath different people could be a result of geographical effect or confounding effect. And therefore, in order to be careful with our analysis, we have taken with every category of these diseases a control group, namely a person that is healthy in the same places. And we have compared between the healthy, it's assigned here by C, between healthy people between different countries and between different clinical centers. And as you can see, there is no difference between the people that are healthy, disregarding of their geographical place or genetics or food habits, etc., etc. And therefore, we conclude that these results are real results and not as a result of confounding factors. The easiness of this device that you have a hardware such as the smartphone, which I handle, but you can always use it always differently by tailoring the artificial intelligence or the machine learning in a proper way remotely. This can be really very helpful for a wide variety of applications, including in pandemics. In February last year, before we had the first case of COVID-19 in Israel, our eyes were already in Wuhan, in China, where we have sent our devices by FedEx, as you can see here. And the medical team has received our device. And then from remotely, we have controlled the algorithm which operate the devices, and we have investigated which kind of sensors can really work for the COVID-19, because this is our first experience with this type of disease. And within a very short period, we have done very big clinical study, and then we have expanded it to more than 5 clinical centers worldwide. And as you can see, the person went into the clinic, excelled into the device, the data was broadcasted to us, and we have done the analysis. Part of these results could be seen here on the screen, where we have shown that if we want to detect COVID-19 and to differentiate it from control groups, namely people that has no signs for COVID-19, we could get a sensitivity of 100%. In this case, the most important part is the sensitivity, both in the training and the test set. But the other challenge was that we are close to the winter, or we will be in the winter, and there will be a lot or many kinds of influenza. And therefore, there is a need to distinguish between COVID-19 and also infection disease that are not COVID-19, which I assign here by lung infection. And therefore, we have done more study on this case, and we have received sensitivity of 97% and 100%, where the specificity was lower. But for the purposes of the screening, we can rely on this result, which is pretty good. Furthermore, throughout breath analysis, we have asked the question whether we can monitor people throughout the curation or their treatment from a disease. And therefore, we have taken a group of people, and we have made a follow-up by breath analysis, and the results also were quite successful. So in every case in this device, we use the same set of sensors, but in every case we choose which specific types of chemistries we apply for the specific application. Namely, we tailor the algorithm and the sensors as a toolkit to the kind of the application that we do. And as you can say, with this tailor-made approach, we could be successful with this approach. This was published also in the last year. The breath analysis technology was pretty promising technology, but one short story that happened to me in one country, that I have seen and understood and realized before 6 or 7 years, that even though the simplest technologies that we use in the Western countries might be really complicated to people and populations in the developing countries, who live on less than $1 per day and mostly disconnected from infrastructure of electricity and infrastructure of water. And therefore, the breath analyzer, which I have shown you, even sometimes might be challenging to be used in some countries in the developing areas. And therefore, we have thought, why we don't develop an approach that could be more adaptable and could be accessible to those populations. And therefore, the idea was to take the same technology of the sensors that I have shown earlier, the same concept, but to build it in different way, to make it on a thin film of device, as a wearable device, as a tattoo that you can put it on your skin. And it will detect the volatile biomarkers of the disease that are emitted from the disease into the skin. Of course, back then, before 7 years, when we have started our collaboration with the Gates Foundation, and later on Bill Gates by himself got into this project. There was no evidence in the literature that the skin has a unique fingerprint of infection in the way that we wanted. And therefore, we have done a huge amount of research with blind analysis in many clinical centers, where afterwards we have provided the proof that skin in many instances could be served as a source for biomarkers of a disease. Next week, it will be released in Science Advances, this article, which talk about the, the proof per se. So the idea is to make a patch with the same concept which I've spoken about and to detect the disease. But nevertheless, we have another challenge in the developing countries, that if you use a patch and this is very thin, it might be scratched, or on the other hand, it might be cut. And therefore, to replace it, even though it costs $1, it will be a real challenge. And therefore, we have thought about how we can induce more sustainability of the device in case it cut or scratched. And therefore, we have thought about building the whole electronics on the device, or at least the main components of the device, such as the electrodes, the channel, the dielectric layer, etc., by a self-repairing approach. Namely, even if that will be cut, the device will repair itself in autonomous way, without temperature, without UV, etc., etc. So this is more or less the idea. Again, there are many details. You can read about them in the articles, but what we have done at advanced stages of our study, we have succeeded to build a tattoo-like device with a thickness of 200 micrometers that includes, in this case, 5 field effect transistors. And I will speak about the field effect transistor within a minute, how it's built, very simple. And this field effect transistor has already receptors and functional groups on the inner side, which is in touch with the skin and can detect the volatile biomarkers which are appearing from the skin itself. The way we apply this device or this patch on the skin is very easy and similar to the tattoo which we put for our kids, or they put for themselves, more accurate. So we wet the device with water, and we peel off a paper-like substrate, and the device will be on the skin. And that's it. Of course, there is a need to connect it either throughout RFID or NFC to electronics, or to a way to transfer the information. But the main core, it's this core. Of course, it can be stretched and aggregated quite nicely. And I will speak about this property within a minute. So this device practically is built from 3 different layers, because it's field effect transistor. The first layer is the dielectric layer, which based on polyurethane after some modifications. The electrodes of the device of the source and the drain are built from polyurethane, together with carbon nanotubes that are highly conductive, something that we can separate in the lab, as you know, where the conducting channel or the semiconducting channel of the field effect transistor was built from a mixture of polyurethane together with semiconducting carbon nanotubes. And together we have printed that onto a device where you can see here the behavior of the device or the field effect transistor. You can see the drain current versus the drain voltage and versus the gate, where the performance is not that bad for a sensor, even though much better performance can be, of course, achieved. The device, as you can see, has received exactly what we have predicted or what we have wanted. As you can see, if we scratch the device then after 12 hours, you can see that the scratch will be start to eliminate step by step. After 24 hours, the scratch in autonomous way without any intervention close the scratch and really heal in 100%. Of course, as you can see here, if we cut the device, we change the conductance, but as you can see, the device goes back to its normal level. And the more we wait, the more it goes to its original value. Here you can see the device, the original device in the black curve. We have damaged the device. You can see in red color how the device behaved. Of course, we don't see almost any behavior of the field effect transistor. After 10 minutes and after 30 minutes, we have improvement, and the device goes back to its field effect transistor behavior. So this is something that you can see that we could heal not only the mechanical aspects of the device, but also the conductance and even the device behavior in terms of electronics. Of course, with this technology, we have repeated the experiment many times, but after the 5th time, we started to see challenges with this approach, and we have started to see after the 5th time some drifts as a function of time. In this case, you can see one of the most important parts of this device, thanks to nanotechnology, that if you stretch the device by 50%, which is really very significant, okay, you don't see almost a change in the field effect transistor. And this is very important because most of the wearable devices which we have today in the world are affected by movement or stretching. And therefore, to find a technology or a device that is not affected or insensitive to pressure or strain, but still give you the same behavior of the device performance, that will be absolutely great. You can see that we have tried this stretching back and forth 200 cycles, and you can see that in many cases we got at the first got very nice results and repeatability and reproducibility with this device. And as could be seen here, the device could work on detection of wide variety of temperatures, also on pH, again, by defining the chemistry on the surface. But the most important part of the self-healing that you can always compare between the black color and the red color, in this case, namely before cut and after cut, or the comparison of the self-healing performance. And you can see that once the self-healing of the polymer and the device and the electronics on the surface is accomplished, you can see that we go back to the original status. So there are many examples here. And of course, this self-healing property was proven to be good between 0 to 60 centigrades, which were quite nice. Nevertheless, with this approach, with this device generation, we had some challenges. One of these challenges that the device, which I have shown you earlier, despite its good practices, it worked only in gas phase. At the time, we might need also to use the device also in aqueous solution or inside a liquid for whatever applications that you have in mind. And therefore, one challenge is how we can operate that device and how we can induce the self-healing both in the gas ambient conditions and also water, and not pure water, but also a water that includes a lot of contaminations and a lot of salts and other components. The other issue, how we can reduce the time of the self-healing from 24 hours to a few seconds, so it will be much more practical. And the other challenge is how we can combine on the same surface of the device many functional groups that could enable us to detect wide variety of parameters simultaneously, whether physical parameters or chemical parameters. And in the next few minutes, I will show you how we have done so. So as a first step to reach this ultimate goal, we have introduced a new self-healing thermoplastic elastomer for use under harsh and complex humidity and also under water conditions. And more specifically, we have made a synthesis of a new polymer from hydroxyl terminated polybutadiene that as you can see, is extended by isophron diisonate and 4-aminophenyl disulfide. And we have done it by 2 reactions. And in this structure, the polypeptidane parts of the fourth elastomer contributed to the low temperature flexibility and to the water resistance, where the 4-aminophenyl disulfide component contributed to the dynamic disulfide and hydrogen bonds when the urea and the urethane linkages are formed. So we rely here on hydrogen and disulfide bonds in order to do the self-healing process. And of course, the integration between the 2 integrated mechanisms of the self-healing were based on the covalent disulfide and non-covalent bonds of the hydrogen. And also in parallel, we have to take care about the softness, and therefore a high softness was also achieved by the balance between these components in this case. We have done simulations of molecular dynamics, and we have shown that there are 2 regimes in this structure, some of which are soft and the other part are hard, which you can see here also from the TAM image, also from these schematics. And the simulations showed that the water molecules can only interact with the hard regimes, and they barely diffuse into the soft regimes in the new self-healing polymer, which you see here on the screen. And the hard regimes don't form a continuous pathway into the polymeric bulk. And with this approach, we could really develop a device that works underwater without being affected by these results. Here in the top left image, we show that the polymer can recover its elastic properties after a complete cut. And the right side images here shows a full generation of the scratches within 24 hours under ambient conditions. We see also similar behavior towards the cuts underwater. Again, all of these are underwater conditions, which is really very challenging. And of course, you can see that it works quite nicely under a wide variety of pH groups. And here you can see that the results are not affected by the pH, more or less within the statistical error is not affected by the pH and the conditions of the water in which the healing is being done. I hope it will work. Okay, not this one. So just to give you a demonstration of the device. So this is the polymer that has been developed. And this is a water from the Mediterranean Sea, from Haifa beach. And as you can see, we have cut the device. We have reconnected the device without any intermediates or glue. And within a minute, you will see that this device, okay, I will speed it up. After this device it revived and regenerated its mechanical strength very nicely and can hold quite good mechanical aspects. So, in terms of the electrical performance, the device, as you can see, really can work and operate this LED. And right now, we will not scratch the device, but we will cut it in the middle. And you can see that the device stopped working. And after we have just brought the 2 parts together, the device started to blink back. I will speed it up, blink back, which means that even the electronic behavior started to go back into life. This device, and I will go here without going into more details because of the time limit. This device, after combination of different chemistries on the surface, we have included a device that include pressure sensors, pH sensors, temperature sensors, and devices that can also detect glucose, cortisol, pH, salt on the same device itself. You can read about it in this article. And even though you cut the device from the middle everywhere, even in this pH sensor or temperature sensor, you always get back to the same performance after the healing process. This is an example what happened for the temperature sensors, and this is what happened, for example, for the pressure sensor. And as you can see, the differences between the black points and the red points, which indicate the before healing and after healing process, is exactly almost the same. The good news that we have repeated this experiment 1,000 times, and we got deviations of ±5%, which is very, very good results. So recently we have also developed, we have integrated heaters inside or under the layers of the self-healing device in order to speed up the healing process. And then we could, with the heating specific areas, by a mechanism of neural network, to make the healing process not within 24 hours, but within 30 seconds, sorry, only. And with this device, as you can see, again, with the chemistry, we could detect wide variety of compounds. The most important part before my next two slides and final two slides, that this device also doesn't only revive its electrical behavior and mechanical behavior, but also chemical sensing behavior. And this is an example that we have taken biomarkers from the skin before any cutting process, and the device could detect wide variety of compounds, as could be seen here, with different concentration, and separate between them very nicely. We have cut the device in the middle. We have reconnected the device. We have waited 1 day. And as you can see, even after 1 day, the device still performed quite nicely and could differentiate between the biomarkers and their concentration in very significant way. Of course, if you take a deeper look into the device, you can see we have some deviations here and there, but the overall observation that we could revive the work. The final thing that in order to bring this device not only within lab conditions and not only as a proof of concept, we have taken this device also into real clinics in the developing countries, in South Africa, in India, and in Latvia, on 1,000 patients that have tuberculosis, which is a quite significant infectious disease in the developing countries today. And we have tried to put the device on the skin and to make the detection. The analysis were done by Microsoft Seattle because they have a relation to the funder who funded this project. And their analysis have shown that we could detect the tuberculosis, namely the infection disease, and discriminate it from healthy controls by 92 to 97% accuracy. And furthermore, we could discriminate between the infectious disease and other infections that are not related to the tuberculosis, also with similar accuracies. In very big cohort, we have also tried to discriminate with this device, which is a simple device, as could be seen, between the what is called latent infection disease and apparent or active infection disease, namely infection disease that is found in the human body, but you don't see any side effect or you don't feel it. And infection disease, the active one, that you have it inside your human body, but you feel the side effects. This is very big challenge, but we could get an accuracy of 88% to 94%. These are absolutely excellent results, but of course, always in the clinical fields, there are always a need to try more geographies, more people, and more heterogeneity between these people, and more. So I'm in time, and therefore, just to summarize my talk, with a proper design of materials and nanotechnology and integration with the machine learning, we can build a device as a toolkit that we can tailor it according to the application in very easy way. Sometimes you can use the same sensors, but with different algorithms, you can operate the device differently, and this will cost a lot of challenges and a lot of infrastructure. And therefore, relying on artificial intelligence that are integrated with the devices could help us to increase the sensitivity or to compensate for disadvantages that we might have in the hardware per se that we cannot otherwise overcome in other technologies because of some challenges that we have in the field. Finally, the team who do this work here on the screen. So thanks for my team members for their hard work and for you for your attention. Thank you.
Okay, thank you, Hossam, for this very amazing talk. And guys, we have time for discussion. Any questions?
Can I ask, Shachar?
Sure.
Okay. Hi, Hossam, really very, very interesting. I want to ask about the first part, the basic feature of the sensor. Can you give just an example, a simple example of how it works? Is it a molecular recognition, how it is translated to an electrical signal and so?
Absolutely. Just a minute. Still, I prefer to show it to you by this slide.
Okay.
1 minute. Okay. Okay. So I hope you see the screen. Okay. So first of all, we don't have any chemical reaction in this case. So this will allow us to make the recovery of the sensor. So we have gold nanoparticles in this case, as could be seen here, which are coated with organic ligands. And the organic ligands have a functional group that don't react chemically with the other functional group on the other side of the nanoparticle. So if you have one nanoparticle and the other nanoparticle, they are separated by the ligands, but they don't react chemically. And therefore, if you build a film of these nanoparticles, the molecules will penetrate throughout these nanoparticles inside, and they will do one of three mechanisms. One mechanism is called aggregation, namely the molecules from the gaseous phase, which penetrated throughout the porous or spaces between the nanoparticles, will bring together the nanoparticles close to each other, and they will do a mechanism which is called aggregation. In terms of morphology, you can see here in the green color, it's aggregated. During this aggregation, the conductance will increase quite significantly. And by measuring the difference between the conductance before and after, this will be one mechanism to make the detection of these kind of molecules, which have high affinity towards the ligands on the surface. On the other hand, you might have other issue. We have the other molecules and other chemistries. When they interact together, the film will swell and do swelling. And based on this swelling, they will increase the distance between the molecules and the conductance will decrease. And of course, we measure decrease in the conductance. And by that, we can get our sensing signal. The other mechanism is the dielectric mechanism. If we have molecules with high dielectric layers, such as water, so they just go inside and they control the whole mechanism throughout the transfer, throughout the medium in a quite proper way. So this is on the basis of the sensors, and there are many subcategories of the mechanism itself. But in our approach, we cannot rely on one sensor because it's not selective. And therefore, we usually use a wide variety of different chemistries, and we compare between all of them together with one number. That this number is being given by machine learning by some mathematical models which project the multi-dimension to single dimension, which we can and allow us to make this comparison.
Very interesting. And if you want to refresh the sensor, to take a second measurement, what do you do?
Absolutely. This is because we don't have a chemical reaction, the refreshing the sensor can be done by applying a small, short vacuum. Sometimes we can use a few hundreds of milliliter, or you just blow it with fresh air or fresh nitrogen.
Okay, great. Thanks.
Thank you.
Okay, more questions?
Yeah, I have a question, Hossam. I'd also like to thank you. I'm really fascinating. And actually, two questions, if I could. One is you have all these specific sensors, but you have very complex mixtures. And to what extent in some of the mixtures do you have to worry about interference of one component with another, either enhancing or reducing the sensitivity to that? And then the other question after that is just maybe a technical one. I noticed when you were talking about the COVID, that for the specificity, the column of the specificity that COVID cured, the testing set did better than the training set, which is really strange behavior.
Mm-hmm.
Yeah.
Okay, I will answer the first question. Of course, in a given breath or a given mixture, a complex mixture, we have hundreds of compounds inside the breath, and we have also water, which make complexity. So because we use a limited number of sensors, we don't detect every compound in a specific or selective way, but rather we take the whole mixture, and the whole mixture will apply on the surface of the sensor. And we take the overall mixture. But sometimes a given compound, as you have said, will change or have a variance between one patient to the other patient. And in this case, I will try to explain it in a very simple way. The single biomarker is at the range of few ppbs to tens of ppbs, but the whole mixture, if you sum up the concentrations together, it will be up to few tens of ppms or hundreds of ppm. And therefore, a change or variance of one biomarker or one compound within this whole mixture, few ppb out of few ppm, will be negligible, and therefore it will less affect us. Unless otherwise that biomarker really get into very selective and very dominant interaction with the device itself. And it happens sometimes. And therefore, in many cases here, we always try to correlate between the confounding factors, and we do mass spectrometry in order to see whether we have any strange behaviors in this case. For the COVID-19, of course, the blind analysis in some instances were higher than the training set. And I agree with you, this is strange, but again, this is blind analysis. So we have to rely on it. And if you take a look on our article, we have warned that the population in that specific case was small. And of course, we have a risk in this case of the analysis itself, but this is a matter of the size of the population, which we have warned about it. And today we have extended this experiment to more than 1,500 or 700 patients. And I can tell you the blind analysis is lower than the training analysis with a few percents, and things looks much better. So I can blame in that case is some overfitting because of the small population.
Great. More questions?
Actually, you're hearing me?
Yes.
Okay, actually, Professor Hossam, I send you on chat the question, but I can also say it here. Again in Hebrew or just in English?
You can
As you
speak in
wish.
Hebrew, it's okay.
אוקיי, אז השאלה שלי ככה, אני לא יודעת אם ציינת את השאלה הזאת או ענית על זה, אבל האם הסניף ההונאה האלקטרוני קיבל אישור מה-FDA? למרות שאחוז ההצלחה זה מאוד גבוה, נכון?
כרגע הוא מקבל אישור מה-CFDA, וישנה חברה אחרת ישראלית שנקראת נאנוז מדיקל, שכרגע משמישה את זה שוב ל-COVID-19 ועוד מחלה אחרת, אבל שוב, כיוון שזה עניין של שנה, אז הם בפרוסס עצמו. אבל בינתיים ישנו CMI.
זה עדיין לא קיבל, בגלל זה אנחנו לא משתמשים בו במרפאות, כאילו לא רואים אותו בבתי חולים, במרפאות וזה.
לא, קיבל CE מרק, אבל שוב, ה-COVID-19 הייתה הפתעה די גדולה, אבל במחלות אחרות הוא קיבל את ה-CFDA.
CFDA. זאת אומרת שזה אחרת מה-FDA?
לא, CFDA זה Chinese Food and Drug Administration, קיבל גם כן את ה-CE Mark, שזה שוק די גדול, את ה-CE Mark, שזה באירופה, ב-FDA זה בפרוסס, זה הרבה יותר ארוך.
בסדר, בהצלחה.
תודה.
Okay, more questions? Okay, if not, so really thank you, Hossam, and good luck.
Thank you. A pleasure.
Okay.
Have a nice day.
Thank you.
בהצלחה ותודה.
תודה. ביי.
תודה רבה.